for our microscopy lab tomorrow, okay, the purpose of the microscopy lab is not to be able to identify all the little structures inside of the 3 microorganisms that we're going to be looking at. Okay? The purpose of tomorrow's lab is to gain experience using the microscope and learn how the microscope itself works. Okay? As well as gain some experience with some of the techniques associated with using the microscope, like making a wet mount, okay? Using a microscope slide, okay? Things of that nature. All right, so those are going to be all the things that we're going to be working on tomorrow. So the problem we're investigating with this lab is, how does a microscope allow us to see tiny specimens? Well, we talked a little bit about that yesterday when we talked about how Hooke designed and built the first microscope. He understood that a lens was going to alter the path of light coming from a specimen. He could alter that path of light in such a way that he could make the light diverge. He would make the object look bigger. That might be something about how a microscope works. You're welcome. Okay. In this lab, we're going to be looking at 4 different specimens. Okay. But we're going to be doing that with the same microscope. And we're going to be looking at how they appear under different magnifications. Okay, we're using all the different settings and things like that on the microscope because we're going to be looking at different specimens. Okay, we're hoping that those different specimens are going to look a certain way when we look at them through the microscope. Incidentally, I'm talking about the design part here, but I'm being much more cryptic about it than normal. Okay, trying to wean you off of giving you the variables directly, even though I'm still kind of doing that. Okay. Now, things that can affect how a microscope works need to be controlled each time I look at a new specimen, hoping that it will look a certain way because it's being magnified by the microscope. Those certain things are usually going to be light related. Okay? They are not really going to be environmental. A microscope will work whether it's 30 degrees or minus 30 degrees. Okay? It won't work. The knobs won't turn as well. Okay? But the whole light focusing part will work just exactly the same. So you don't have to put in temperature as a controlled variable. It's not. Okay? But anything that has to do with the path of light or how you view the specimens or even maybe how you take their picture. Okay? Because that affects how they appear. Okay? And we're worried about how they appear. Anything like that could be considered a controlled variable. Now, for our hypothesis, okay, the problem we're investigating is how a microscope works. So the if part should probably talk about that. Works. Okay, the and part, as always, describes the experiment. Microorganisms are viewed under a microscope. Okay. The then part, as always, predicts our findings. So I want a prediction here about how the specimens we view under the microscope are going to appear. I'm looking for more than bigger. Okay, I'm just putting that out there. In terms of level of detail, I am looking for more than then the specimens will appear enlarged. And, okay, a little more detail. Maybe a little bit about why and why. Okay, so yeah, put some stuff in there about the appearance of the specimens. Okay, when we're in the lab tomorrow. Okay, the procedure is pretty straightforward. You're going to go to the back cabinet. I'll have it open already. You're going to grab a microscope, carrying it with 2 hands, back to your table. Okay, it says here, get the necessary slides. But I want to add to that, not all at once. Okay, you only need 1 specimen on your table at a time, because that's all that you can look at under the microscope at one time. Okay, and what I find happens is if there's lots of microscope slides on the table, they tend to get brushed off by accident onto the floor. Okay, so 1 specimen at a time. You will be viewing these specimens in a particular order. Right? That is, you will be looking at the letter E first, the amoeba second, the Paramecium third, and the Euglena last. Okay, the reason is we're going from biggest to smallest. Okay, we want to look at the things that are the easiest to find and view and focus rather than looking at the things that are much smaller. Okay. So the letter E is not a prepared slide. You are going to have to make this slide. Making a slide is called making a wet mount. Okay? Making a wet mount works like this. So a microscope slide is just a long rectangular piece of glass. Okay, so you will have one of those. You will have at the front of the room several pairs of lab scissors and some newspapers. You are going to take a lowercase letter E from an article, not a headline, okay, so it's nice and small, and you're going to cut it out. Right? You're going to take that little piece of paper, okay, with the letter E on it, back to your table. Okay, and you're going to put it on the slide, somewhere near the middle. Okay, then, okay, you will have two other things. You will have a cover slip, which is a square piece of transparent plastic, and you'll have an eyedropper with some water in it. Okay? Once you put the letter E on the microscope slide, you want to make sure the letter E is right side up. Then you want to put the eyedropper on the letter E, like literally pushing it onto the slide, and dispense some water. If you drop the water onto the letter E, it'll flip over. Okay? Because it'll use those properties of water, right? And it'll just ride on top of the bubble. We don't want that. Okay? Put it right on the letter E and slowly push some water out so it soaks it, and then the water kind of sits on the slide. The letter E is kind of stuck to the slide. Okay? And you've got basically a bubble of water around the letter E. Okay? Everybody with me there? All right. Now, looking at it from the side, here's my slide. Okay? This is my piece of paper with the letter E on it. I'm going to take this cover slip and I'm going to set it on the slide at an angle like this. And then I'm going to let it go so that it falls down onto the letter E. What it'll do at that point is it'll squish the water and stick to the slide. That's basically the whole reason we use water, make a wet mount. We could make a dry mount, but what would happen to the letter E in the cover slip? They will probably fall off. So water does something that's kind of important here. It might even be important for one of the analysis questions if you were looking ahead. I saw a few people clicking on their keys because they looked ahead to the questions already. Okay, so the cover slip will fall down onto the letter E and it'll sit there. You want to make sure when you put your now built wet mount under the microscope that the letter E is still right side up. Okay, because it should look different through the microscope. But if you put it in already upside down, it's not going to look right when you look at it. Okay, so make sure it's right side up for you when you look through the microscope. Okay, so that's how you make a letter E. I'll go over that, or wet mount of the letter E. I'll go over that again tomorrow, but that's the first step. Now, the letter E, and you may want to make a note of this, you will take a picture of it on scanning power. So your lab diagram for the letter E will be done on scanning power. Once you have the picture of the letter E, okay, got through your microscope and all your group members have seen it, okay, and I would advise here, guys, that we're going to be in partners. Okay, so everybody gets a chance to use the microscope. One partner can do the focusing for the letter E, the next partner can do the amoeba, then the next partner can do the paramecium, and so on, so everybody gets a chance to use it. Okay, once you're done with the letter E, take that slide off of the microscope, put the cover slip and the letter E in the garbage. Okay, the cover slips are disposable. Right, so they go in the garbage, the letter E goes in the garbage, wipe the microscope slide down with a paper towel, put it back in the box. Okay, there'll be a box at the front. Then you're going to move on to the prepared slide. So I'll have boxes of slides up there. One box will say amoeba, one will say paramecium, one will say euglena. Okay, you're going to get the amoeba next. If you hold the amoeba slide up to the light, you can actually see the amoebas with the naked eye. They're going to look like a little purple speck, okay, on the slide, so you'll know what you're looking for. Okay. You'll put the amoeba under the microscope, and you will find it on scanning power, focus, center, go to low power, focus, and center. You are going to do your lab diagram of the amoeba on the low power lens. It is a fairly large microorganism. If you try to get it on high power, it'll take up more than the entire field of view. Okay? The other problem with the amoeba is, because it's so large, it's actually got enough depth that you can't focus the whole organism at one time. Okay? The problem with it is this. Right now, I'm looking at Colton. Colton is in focus, Elise is not. Okay? That's the way the human eye works, right? If I focus here, everything behind the depth that I'm looking at looks out of focus. The same thing will happen with the amoeba on high power. You'll get like the closest part in focus, and then the rest of it behind will be out of focus, and you have to like focus through. It's hard to get a good picture of it when half of it is out of focus. Okay, so you want to take a picture of it on the low power lens because you'll see the whole thing and be able to get the whole thing in focus. Okay, once you've got your picture of the amoeba, go put the amoeba slide back in the box. Grab the paramecium slide, come back. Paramecium, that one you're going to need to get on high power. It's a bit smaller, but still fairly easy to find, and there's lots of them on it. Okay, on each slide. There are different dye lots for those. Some of them, they're all blue, and some of them, they're like some blue ones, some green ones, some brown ones, and some orange ones. And it actually has to do with the genders. Okay? Genders. Many. Paramecium have, this weird thing going on. Okay? Where they can actually reproduce asexually by splitting in half, but they can also reproduce sexually by exchanging a small micronucleus that they have in addition to their main nucleus. So if there's lots of paramecium around, they'll make these little micronuclei and they'll trade them. Okay? And then they'll merge with their main nucleus, and they will be genetically altered, and they'll produce non-clones of themselves. Okay? That's kind of the drawback to asexual reproduction. Asexual reproduction, you just make an exact copy of yourself. Okay? It has all the weaknesses that you have. Right? If you're using sexual reproduction, then you get different combination of genes in the offspring, and that's good. That increases variation. Okay, so that'll be for the paramecium. Then once you've got the paramecium, picture taken on high power, you'll go get the euglena. The euglena is really, really small. Okay, it's really, really small. Under scanning power, you will barely see it. They will be tiny little blue flecks, like a little sliver of very light blue. Okay, you'll need to get them on high power, and even on high power, you're not going to see a whole lot of detail. They have a similar shape to the paramecium, but they're much smaller. Okay, so just to give you some idea what you'll be seeing. Okay, I talked about all this stuff. Okay, here we go. So the amoeba, I already showed you one picture of an amoeba. This is another amoeba. Just like snowflakes, no two amoebas look the same. Okay, and you can see here, there's a bit of depth, right? The front of it and the nucleus is kind of in focus, but this little pseudopod and this pseudopod back here are out of focus. Still part of that amoeba, but it's got a lot of depth. Okay, the paramecium is kind of oblong, okay, almost hourglass shaped, okay, and it's got around the edges here, you can kind of see the little cilia. They look like little hairs, but that's how it moves. It waves these little cilia around. Okay, a euglena, this is greatly enlarged. You'll never see it this big. Okay, but the euglena has a similar shape, but it doesn't have cilia. It has one long whip-like flagellum that it uses to propel itself. The cool thing about a euglena is it uses photosynthesis for energy, but it moves around. So it's a moving plant, kind of. Okay, yeah, it's a very unique organism. Right? That's why it's so green. You can see the chloroplasts everywhere inside of it. So those are the 3 microorganisms you'll be looking at tomorrow under the microscope. Okay, but yeah, the euglena is a lot smaller than that. Yes, these are prepared slides. They've been sealed with resin for, probably longer than you've been alive for old slides. Yeah. Okay. Okay. Questions on that for tomorrow? All right, now let's quickly talk about the analysis. That way tomorrow we've got all class period for the lab, except for the quiz that you'll have at the beginning of class, which will post later. Okay, so for analysis here, using what you saw under the microscope, size, orientation, and general characteristics, explain what you think happened to enlarge the specimens. This is probably where you should talk about that whole thing with the lenses and the light. Okay, I want an explanation. I saw that the letter E looked like this, and here's why. Okay, I saw that the amoeba looked like this. Here's why. Okay, but basically, it's the letter E that's going to help you the most with your explanation here because you know what a letter E is supposed to look like. Okay, and it's going to look different under the microscope, and you've got to explain why that happens. Okay. For question number 2, describe and explain the steps you took to find and focus your paramecium. Basically, I want you to walk me through. First, I put it here, then I did this. Walk me through the steps you used to get it there, just as a review of here's how I get something under focus under the microscope. And question number 3, why do we add water to a wet mount? We kind of talked about that. I would see if you could work some of that polar nature of water stuff into that answer. Okay, could be important, hint, hint, it's important for the answer to that question. Okay, and then conclusion, same as always, copy paste your hypothesis, explain using especially the letter E, but also some of the others, why you can accept your hypothesis about how the microscope works. Okay, and for your evaluation, sources of error. I'll give you one. Inexperience. Okay, you've probably not used a microscope all that much in your life. Okay, as a result, you're not going to be all that good at it tomorrow. Okay, and that's okay. You're going to learn a lot. It's not rocket science. It's biology. Okay, so you'll be able to pick it up no problem. It's not physics, it's biology. Okay, so we'll get you all experienced with that, and you guys will be able to use the microscopes really effectively. Then you have to come up with one more. I'm sure you'll figure one out tomorrow. All right, questions on any of that? Okay, so you still got like almost 15 minutes here. You can probably get your design and hypothesis and stuff ready to go, okay, for that.